Hi, I'm Ivy Shu. I'm going to be a junior at Hudson High School. I did a science project in the science internship program with Dr. Takuya Sakaguchi, and my project was the molecular genetics of non-alcoholic fatty liver disease. So the implications of non-alcoholic fatty liver disease is a widespread condition that affects up to 25% of the people in the United States. It's the buildup of excess fat and lipids in the liver, which can lead to hepatocellular carcinoma, commonly called liver cancer. Hepatocellular carcinoma is the fifth most common cancer and the third most common cause of death from cancer worldwide. And the mortality to incidence ratio is 0.93, so obviously it's very deadly. NAFLD is also commonly associated with diabetes, high cholesterol, and high triglycerides. My project focuses on the earliest stage of NAFLD called steatosis. Steatosis is characterized by deposition of triglycerides as lipid droplets in the cytoplasm of liver cells. If 5 to 10% of the liver's weight comes from fat, it is said to have steatosis. It is not deadly alone, but it has a high potential to develop into deadly forms. Steatosis can develop into steatohepatitis, which includes swelling of the liver, some liver damage, abnormalities, and death. Steatohepatic cells can also further develop into cirrhosis, scarring of the liver, and ultimately hepatocellular carcinoma. Our lab created a mutation called the S850 mutation to study non-alcoholic fatty liver disease. The S850 mutation is a mutation in GMP synthase that leads to a phenotype of steatosis in zebrafish models. When the GMP synthase can no longer produce GMP normally, the fish develop steatosis. We are studying the probable causes of NAFLD through genetics. De novo lipogenesis and lipid transportation are both probable causes of NAFLD. De novo lipogenesis is the formation of new lipids within the cell. Lipid transportation involves the cell absorption of fat and the movement of lipid in and out of the cell. Lipid oxidation is also a probable cause of NAFLD. It is a process of metabolizing fat in the liver and is often referred to as beta oxidation. It occurs in the mitochondria of the cell, and each round of lipid oxidation produces 1 mole of NADH, FADH2, and acetyl-CoA. The end product, the acetyl-CoA, enters the TCA cycle so it can further be oxidized into CO2. So our hypothesis was that we hypothesized that genes involved in de novo lipogenesis, lipid oxidation, or lipid transportation will be altered in S850 mutants. To study this, we did qPCR. qPCR is a way of measuring gene expression levels related to the de novo lipogenesis, lipid oxidation, and lipid transportation and uptake. qPCR is called quantitative polymerase chain reaction and occurs within a qPCR machine. It amplifies the DNA and simultaneously quantifies the amplified DNA segment. The process relies on thermal cycling or repeated heating and cooling. First, the DNA is denatured and the double strands are separated. Annealing then allows the primers to attach the single strands of DNA. DNA polymerase brings nucleotides to make the DNA double-stranded again in elongation. A fluorescent dye, in our case we use cybergreen, binds to the newly formed double strands of DNA. The intensity of the fluorescence is directly proportional to the concentration of DNA product, so the intensity is measured after each amplification cycle. When this intensity is significantly above background levels and past the threshold, the cycle value is recorded. So for our qPCR, we used 3 biological samples. These samples were prepared by extracting total RNA from the S850 mutants and their wild-type siblings 6 days post-fertilization, and then converting this RNA to cDNA. The gene RPP0 was used as a control based on studies that have shown that gene expression is consistent in mutant and wild-type zebrafish. The data was analyzed using a one-tailed paired t-test, and p-values below 0.05 were declared significant. Our results showed that expression levels for genes related to de novo lipogenesis were not strongly up or down regulated. As you can see from the graph, the tested genes show no clear trend in up or down regulation, and only 3 genes had statistically significant expression values. However, the expression level for genes involved in lipid oxidation were found to be strongly downregulated. From the graph, the genes showed a downregulated trend. Furthermore, all genes were statistically significant. Expression levels for genes related to lipid transportation was not statistically significant, but the expression level for the gene related to lipid uptake was downregulated. If de novo lipogenesis had showed an upregulated trend, our results would have indicated that it would be a likely cause of steatosis in the S850 mutants. However, de novo lipogenesis had few statistically significant genes, and it showed no clear up or down regulation trend. This data suggests that it is not a likely cause of steatosis. Similarly, because lipid transportation uptake was not shown to be upregulated, the data suggests that lipid absorption and intake of fat is not a likely cause of the steatosis. However, it is apparent that lipid oxidation is strongly downregulated in S850 mutants, suggesting that inadequate metabolism of fat in the cell is a likely cause of the fat buildup in the liver. The data suggests that overall, because the hepatocyte is unable to metabolize enough lipids through the oxidation, there is a buildup of lipid in the liver. Further study on this could be done with Western blotting conformation of certain genes. We could also increase the number of biological samples done with the qPCR, or perform qPCR on more genes involved in lipid oxidation. We could also examine more specific pathways involved in the metabolism and analyze the role of lipid oxidation in the context of other diseases. And finally, I would like to thank my mentor, Dr. Takuya Sakaguchi, for his invaluable help and guidance in this project, as well as the other members of my lab who are always there to lend a hand or answer a question. Without them, this project would not have been possible.